Hello everyone. So today I am presenting preliminary analysis of a project that looked into comorbidity risk scores and immune aging markers in women over 45 years old living with or without HIV in the Carma cohort in British Columbia. So as of 2019, an estimated 19.2 million women were living with HIV globally. This number depends on the country, so for Canada this would be 23%, and for sub-Saharan Africa this would be around 60%. In addition to that, about 48% of estimated 1.7 million new HIV infections were among women. As a result, there are younger women living with HIV who have been newly diagnosed and are facing some additional reproductive challenges, and older women living with HIV who have been either newly diagnosed or living with HIV for years. Despite the fact that second population now constitutes the minority of all women living with HIV in the world, both these groups will be facing unique challenges that are related to aging while living with HIV as a result of successful CART, which increases life expectancy in this population. So these challenges include stigma, social support, mental health, accelerated aging, and higher comorbidity burden at younger age. So this project looks into two last variables. And one way to look at accelerated aging is to look at leukocyte telomere length and whole blood mtDNA content, so mitochondrial DNA content. So these two things are both aging markers impacted by chronic diseases. Telomeres in most cells shorten with each division and have been shown to be associated with HIV, and for older people to be a predictor of mortality. Changes in mtDNA quality or quantity, on the other hand, as well as decline in mitochondrial function, are implicated in biological aging and are primarily linked to metabolic and mitochondrial disorders. So despite the fact that people living with HIV are at higher risk of age-related comorbidities, there are limited data describing multiple comorbidity scores for risk prediction in women living with HIV. So in this project, we aimed to compare between women living with HIV and HIV negative control women the risk scores and immune aging markers. Our study sample is coming from the Carma cohort. So there were 585 women older than 18 years old in the Carma cohort, of which 262 with at least one study visit after the age of 45. However, 59 HIV-negative women were excluded because of lack of clinical data, so not even one comorbidity risk score could be calculated for them. So we were left with our study sample of 203 Carma participants, of which 81 were HIV-negative controls and 122 were women living with HIV. For all these women, risk scores were calculated retrospectively, and LTL and mtDNA were measured with qPCR. Interestingly, women living with HIV in our study sample were younger than HIV-negative control women, and the age of excluded controls did not significantly differ from the age of HIV-negative controls included in the study. So in this project, we looked at a number of comorbidity risk scores which look at different organs as systems, such as cardiovascular, bone, liver, kidney, and all-cause mortality. So some of the scores were calculated for all participants, and some of them only for women living with HIV, because the scores were HIV-specific, so they required some HIV-related variables. For each score, the input is laboratory or demographic data, and the output is a risk of a chronic disease or an event. So for example, for cardiovascular scores, the output would be 10-year risk of cardiovascular event, such as stroke or myocardial infarction. The risk scores were calculated for different subsets of participants based on the clinical data available and some specific score requirements. So for example, for cardiovascular scores, 6 women living with HIV were excluded because they already had the event of interest, stroke or myocardial infarction, in their history. Now, demographically, women living with HIV were more likely to be current substance users, less likely to be current alcohol consumers, more likely to be current smokers, more likely to have had HCV or HPV diagnosis ever, less likely to be high school graduates, and more likely to have a lower income. Also, both groups were ethnically different. Now on to the results section. So these scores were calculated for both groups. As you can see on the left, for KROC score, a larger proportion of women living with HIV had moderate and high KROC compared to controls, indicating higher risk for 10-year fracture. Interestingly, for both cardiovascular scores, we saw no difference. So no difference in 10-year risk of cardiovascular disease. For the APRI score, a larger proportion of women living with HIV had APRI more than 0.7, which is a clinically significant threshold, indicating high risk for hepatic fibrosis. Now on this slide, this table contains HIV-specific scores, so this is descriptive data. So women living with HIV, 18.4 of them were at medium to high risk of liver-related death, 71 were at high risk of chronic kidney disease. The 5-year all-cause mortality risk, when calculated for all women living with HIV in our sample, was 10.8%. But when calculated only for women living with HIV who were current smokers, it was almost doubled to 18%. For reference, for a woman who is not living with HIV of the same age, the 5-year all-cause mortality risk would be 4%. When it comes to aging markers, we saw shorter LTL in women living with HIV compared to controls, and we detected no difference in mitochondrial DNA content. So as a conclusion, in our study, despite the fact that women living with HIV were younger than HIV-negative controls, we observed for the scores that we compared between the groups higher risk scores for liver and bone disease and shorter LTL among women living with HIV compared to controls. In addition, for women living with HIV only, 18.4% of them were at medium to high risk of liver-related death, 71% were at high risk of chronic kidney disease, and the median risk of 5-year all-cause mortality was 10.8. This risk was almost doubled when calculated only for women living with HIV who were current smokers. We detected no differences in cardiovascular risk and mitochondrial DNA content. Considering the fact that the risk scores can inform some treatment decisions and prophylaxis, and it is possible that women living with HIV might need this at younger age, further research is needed to determine how to better predict and prevent aging comorbidities in this population. Thank you.